She is a professor of biology at Howard University. Her research includes the study of human-plant coevolution, particularly the influence of phytochemicals on human metabolic effects and evolutionary processes, and in population substructure in people of African descent as it relates to health disparities. Her peer-reviewed research spans the fields of biology, anthropology, and medicine. She is the director of the W. Montague Cobb Research Laboratory at Howard University, the largest collection of African-American skeletal and dental remains in the world, covering 400 years of African-American biological history. In 2017, Howard University named her STEM Woman Researcher of the Year, and she received the Outstanding Service Award from the Department of Biology. The title of her presentation will be Reconciling Past Injustices, Building a Future in African-American Genomics. Dr. Jackson.
I'm very honored to have been asked to participate in this session, and I'd like to thank Rob O'Malley and also Indiana University NAGPRA offices and personnel. I want to say up front that I'm not one of those pseudoscientists who's talking about African Americans are indigenous people. We're not. We were brought here 400 years ago, but because of the intensive, long-standing residential disenfranchisement and the discrimination that we've been subject to, many of the issues that African Americans face are similar to problems that indigenous people have faced. And so finding those similarities and bonds means that some of the initiatives that indigenous people have developed may also be relevant for us in stimulating interest in STEM and so forth. And in fact, that's what we're starting to see. And I find this, the research on cosmology particularly interesting because of its tie-in to mathematics. And I am often teaching students that in fact some of the first mathematicians were probably women because of the need to, because of the obvious synchrony of regulating our menstrual cycles, but also because hunters and gatherers were looking up at the stars so the hunters could figure out where did the gatherers go, where did the women and the children go after they went out searching, tracking down these animals for consumption. Okay, so I want to talk about the Cobb Research Lab and what we're doing that ties into the application of science practice and community engagement, because community for us is absolutely central. We have, at our best, never bought into the Western model of individualism, because the individual doesn't go very far in a racist society. the individual gets smacked down very easily. And so you need the community to support you, to validate you, and to get you through those hard times when everyone else is telling you don't belong. So the Montague Cobb Laboratory, as Dr. O'Malley stated, is the largest collection of African and African American skeletal and dental materials. We have 3 collections, and the third collection from the Rupert's Valley in the South Atlantic came on during my watch. So I'm really proud about the expansion of the collection. And it does give us 400 years of African American biological history. That allows us, that's just the raw material, then we do the science on it, and we use it as a catalyst to encourage student engagement, community participation, and to clarify the nuanced relationships that African Americans have had with the larger society and with each other. And so our guiding perspectives are pretty straightforward, and I really had to think about, what's in the vision that we have that distinguishes us from any other skeletal collection, in the world. And that is that we have tied in spirituality. We see it as a fundamental part of the human condition. So we don't buy into the notion that spirituality and science are on two different planes. They're in fact connected. And part of our discovery and efforts to stimulate ourselves reach back to the spiritual in order to give us inspiration for the science. You know, and I'll be glad to talk about examples of this. But when we moved to our new building, I was going to write for a grant to see if I could get headphones for all of the graduate students so I could pump in some really good music so that they would hear that as they're thinking. Because the creativity is, you know, you have to stimulate it and you have to provide an opportunity for it. And for us, it's tying into things that are culturally relevant, that we feel comfortable with, and that will reveal hidden secrets. And it may be through jazz, it may be through a lecture by an orator or something, but that spirituality, again, is a fundamental part of the human condition and absolutely vital for our creative approaches in science. And that's what we think we can contribute to the world scientific community, is a new way of looking, sometimes at the same old thing, but it's a new way of looking at it and new interpretations. Community engagement is essential, and in fact, we have a very active community so that if we don't consult with them, they simply take us to task and they stop listening to us. So this is, I think, a cultural hangover from West and West Central Africa, where your leader is only a leader as long as he's winning, as long as he's producing things that you like. And if he's not producing and saying things that you like and that you find useful, then the people simply turn away from him. We're committed to applying what we call next generation science, that is the most advanced scientific techniques that are at our disposal now in the 21st century. And we want to do it to all of the collections and then disseminate the results broadly and meaningfully. An example of that is today I just received a message from one of the dental professors, because Howard is really unique. Howard University, it has all of the schools that are on any large campus, but all very close together. So the dental school is across the street. The medical school is across the street and half a block away. You know, arts and sciences is just up the hill. And so what we find is that many of our strongest intellectuals, like Montague Cobb or Ernest Just, Ernest Just was a cell biologist, but he became stimulated and really invented, he was the first to see epigenetics and talk about the power outside of the nucleus of the cell in determining cell function. But he got his inspiration by going up on the hill to talk with the sociologists and the people in the humanities as they talked about civil rights and the society as it was organized to fight the true and full emancipation of African Americans. He said, okay, well, how can I translate that to what's going on in the cell? How does a cell determine who it's going to be and its identity? And he made corollaries with what was being discussed in the social science and humanities communities. Yet he was a cell biologist. So he was able to see what other colleagues in other institutions could not see, because he used those creative forces and he brought in all of the cultural powerhouses beyond simply cell biology. I don't know if that's making sense to people. But it's something that each of us needs to do as well, because science has been defined as something that white men in white lab coats do, and nothing against white men in white lab coats, but they also seem to be kind of asexual and not very interesting people, you know? So I believe that you can do very good science if you bring in and integrate additional sources of stimulation. And then, of course, collaborative interdisciplinary research is emphasized, and interdisciplinary research is very much a part of African American cultural tradition. We really don't make big barriers. In fact, when we make barriers, disciplinary barriers, we usually fall down. But when we start integrating a little bit of sociology, a little bit of anthropology, some biochemistry, some cell and molecular biology, and put it all together, then we start to get more cohesive pictures of what's going on. And that is something that we emphasize a lot in the Cobb Research Lab. So this is just a quick synopsis of the ongoing research. These are the 10 major projects, and I'll talk about the last one in a little bit more detail. But I've got graduate students. So the other thing that we do in my lab is I call it each one, teach one. That means graduate students are responsible for having a cadre of undergraduates that they work with. Undergraduates are responsible if any high schoolers or community people that kind of wander in. So everybody has teaching responsibilities, has leadership responsibilities, and this, of course, frees me because I only have to deal with the graduate students. So there is a plan to the madness, but it creates a hierarchy so that the responsibility is shared. And this is also, it's a part of our culture, and it has allowed us to be way more productive than if I was, hegemonically controlling everything, and they still have to validate what they're doing with me. I'm still the PI in the lab, but I really try not to run it in an authoritarian way because the vision is bigger than what my little mind can come up with. So we have graduate students who are looking at elemental and microbial analysis of the New York African Burial Ground, one of our oldest collections that represents 17th and 18th century Africans and African Americans. Another graduate student is looking at genetic variability and population affinities among the Gullah Geechee and related populations. So the Native American freed populations, the African Americans who were enslaved by Native Americans and then went with them on the Trail of Tears to Oklahoma, those populations also, many of them trace their origins to Gullah Geechee. In fact, there's a group in Mexico, I think they are actually a variant of Muskogee Creek, and they live near the Kickapoo peoples. And they're totally Mexican, except they make sweet potato bread. What Mexicans make sweet potato bread in northern Mexico? No, they came from the Carolinas and then were on their way to Oklahoma and said, no, we think we're going to Texas. And then we're crossing over into Mexico. And they actually worked for the Mexican government for some time and then are still there. But the genetics will allow us to make the reconstructions. Microbial diversity in South Carolina-derived populations, we're looking at the oral microbiome. And we're finding that it's not just the genes, it's not even just the epigenome, that is the chemical markers in association with the genes, but it's also the thousands of microbial species that are inside of us that are also regulating gene expression patterns and making us who we are. So a part of who we are is the traditional foods that we're eating and how those foods have particular patterns of microbes that influence our own gene expression patterns. We're just beginning to understand this, and because the computation of this knowledge requires computers, we've got some computational biologists to help us because we can't just do it with fingers and toes. We need to really kind of run this through larger databases to see what are the correlations. We've published some things on historic tuberculosis in an African American sample, because we have the cause of death for the 19th and 20th century Africans in the Cobb collection. And a good proportion of them died from tuberculosis. Another good number died from cancer. So, we're able to publish on these unique historical patterns of diseases that are pretty common today. But it allows us to ask how far back in time are we seeing these diseases, like prostate cancer, or respiratory cancers. We published a couple of papers on epigenetic consequences of chattel slavery and racial discrimination. And that's really interesting because sometimes you have to use the open source literature because we still have a resistance for hearing the voice of indigenous people or hearing the voice of the oppressed in publications. But my policy has been that if you don't publish it, it's like you never did it. We have a researcher that just published something on addiction in a subgroup of African American youth, and she's been very good at tying in contemporary African Americans with the historical skeletal populations, and so that it stays real for the communities that we try to disseminate our results to. Rib notching is simply when you have extensive high blood pressure, it modifies your anatomy. And so the ribs actually become restructured. So that in our skeletal population, you know, we obviously can't take their blood pressures, but we can look at if we can see if they had high blood pressure in the past, because their ribs will have these little notches on them. We're also looking at the St. Helena's population. These are the groups. These were Africans in the 1830s and 40s who were on their way to enslavement in the Americas, got turned around by the British Navy, dumped at a refugee camp in St. Helena's Island in the South Atlantic. This is where Napoleon was exiled. So as you can imagine, it's a place nobody wants to go to. It's right off the coast of South Africa and Angola. And so these Africans, there must have been almost 30,000 Africans that were dumped there. Like stuff we don't hear about, right? That we never learn in our history. Because our history is very, very Americocentric and even not uniform for all Americans, which is why you have to have these conferences like this, right? So other people are thinking and coming up with models. So the St. Helena's population now has no idea who they're related to. And so we've collected samples from the living population as well as a collection of 325 skeletal deceased individuals whose graves were disrupted when the St. Helena's government decided to build a road, an access road to the airport. So this kind of also gets us back to the whole question of repatriation and whose lives matter. And I'm sure the road could have been built other than through someone's graveyard. But as it turned out, that's what they did, and so we were right on the spot to say, let us have those samples. You know, we want to bring the samples back, and we want to analyze them, the skeletal samples. And then we're spending a lot of time on the construction of African genomic databases. And this is the biggest collaborative project at the Cobb Research Lab. And the big deal about these African genomic databases is that if you have a reference database, then you can compare unknown samples from these understudied populations. So almost all the indigenous populations are understudied. In fact, I can't think of one that's well studied. And African descended people are understudied. And I just saw a report today that came across the internet that said that, you know, the clusters of genes that we use to identify susceptibility to chronic diseases, guess what? They don't work for anybody but Europeans. That they are of limited value for anyone other than Europeans, because even the clusters for chronic diseases like diabetes and hypertension and so forth are population specific. Okay, so I'll go through the rest of the slides pretty quickly. This is our rationale for building that database. We think, yes, it'll be important for African descended populations, but also for everybody else in the world, because everyone's descended from African migrants. Emphasis on African and migrants. Hello, we're all immigrants, you know. Even the Native American people didn't come up out of the ground. They came from someplace else. We can trace it in their DNA and put it in the context, you know, of susceptibilities and resistance. We want to understand African genomic diversity so we can trace human evolution accurately, stop ascribing genes as being European or Eurasian, when in fact they might have their origins in Africa, since we're all Africans. Document the evolutionary process that occurred in the process of migrations out of Africa. So what does it mean to leave Africa, to go through the southern route into Yemen, and then around into India? How does that change your biology? And how do we get the diversity, the polytypism that we see now as a result of long-standing migrations and differential patterns of selection, and also stochastic processes like drift, When we don't have adequate genomic information about continental Africa, then we are lacking adequate information about humans, because our ancestors, our collective ancestors, spent the bulk of our time in Africa. And then we've spent the less time in other places, but we are basically tropical African people. Even when I say that to people from continental Africa, they say, but what does it mean? And I'm still discovering what it means. But I'm trying to look at people who may not obviously look like they came from Africa. I'm looking for the African in you. So when I look at Dr. O'Malley, I'm saying, see now, I think I see some African in him, And I find that this is a way to cut through the notion of, if you look for similarities among humans, then it's a way to cut through the racism we've been taught to see, and the racial categories that we've been taught to see, that we've made far more powerful than they really should be. I said that Europeans were overrepresented in the current genomic databases. So in 2016, about 80% of the people in genetic studies were of European descent, and that in itself is not problematic. The problem is when you try to apply the results of the genetic studies to a decidedly non-African audience. That's where the problem is. But just because, look, most of the researchers are Europeans. People study what they're most interested in. I get that. And they also study what's convenient, which is why in our discipline, how many decades did we spend looking for the land of Eden somewhere in Europe? It's like, really? Why did we do that? Why were there big discussions between the French and the English as to which place could Adam have originated in? Answer is neither. Anyway, one of our colleagues in China said that we know that humans originated in Africa, but we don't know what they did before we left. We don't even know our own history, and that's from a researcher in China. So this recognition, this need for a knowledge of what our past was in our homeland, and our homeland is Africa, continental Africa, that perspective is filtering out into, it's not, what I want to say is, it's not an Afrocentric perspective. It's a human-centric perspective. And once we realize that, then some of the barriers to doing this research begin to diminish. And we say that we can do something about this. Well, this is another rationale, because this is what the data actually look like when we do a chromosomal profiling. This is an African-American woman, and the red areas indicate the genes that are of African origin, of decidedly African origin. The blue are gene sequences that are predominantly found in Europe, and the gray is unassigned. Look at that. The bulk of the information on this African-American woman, this US-born American woman, is in fact unknown. We have no idea. We can guess where those sequences may have come from, but we don't know for sure because we don't have the reference database. So we think that is going to really be a central feature that will allow us to tie together. 3 minutes? Okay. Well, I'll try to go through this quickly. This is just showing the rationale for looking at Africa, because Africa has been studied rather haphazardly. We're trying to look at it ecologically. These are the regions of the world that we're doing our database on: the Atlantic African diaspora, continental Africa, and then the Red Sea African diaspora. And as we collect data, we're already starting to analyze the results. And the idea is we'll produce a reference database, and then we can go back to our skeletal samples, extract the DNA from the skeletal samples, and determine where some of the people in the Cobb Research Lab, some of the skeletal remains, where they actually came from. And can we match people from 400 years ago, 300 years ago, to contemporary populations? For us culturally, as African Americans, that is huge. Because remember, we're a people who have been cut off from our ancestral past. So we don't have our languages, we don't have a lot of our culture, and so anything that will connect us and create that bridge helps to improve our self-image, okay? This is just where we're at. There's a lot of white space here. We're still at the beginning, collecting the 1,000 Atlantic African diaspora genome, the 10,000 continental African genomes, and the 1,000 Red Sea African genomes. And this is just some details on how we've collected it. What we did was we immediately contacted the community and got their priorities, and within an 8-hour stretch, we had collected almost 400 samples. Like, people just lined up and waited for hours and hours to spit in a bottle for us. It was really gratifying. The spit was rolling. Okay. This is just talking about so collecting a sample is one thing. Analyzing and interpreting the data is usually where the hammer of racism really comes in, because the interpretations are based on a set of unspoken assumptions. We're trying to create new assumptions that are based on good history, good social science, good geography, and that's integrated into our models. When that's not integrated in, we get something like this. This is a structure applied to a group of African Americans from Chicago, Baltimore, Pittsburgh, and North Carolina. And Tishkoff reported absolutely no difference between these populations as you read them across, right? But the team were saying failed to find evidence, established evidence of substructure. We know the substructure was there, but their testing was not sensitive enough because it was not geared, it was not cloistered in a culturally relevant context. So, yes, they got some numbers, but they couldn't properly interpret them because they had never studied the people. So here we have, our strategy is discovery 1, discovery 2, and then replication. So we collect this database, we start to see what kinds of correlations we get, we'll do that again, and then we'll try to replicate the findings from the database. This is just showing the community engagement that we did, the community education, local recruitment, then data collection with more community education, because you have to keep talking to the community because they have their own ideas of what is relevant genetically. So you have to keep trying to substitute their ideas with what we understand from the science, but that still addresses their priorities. Where we coded and digitized the data, did data generation and analysis, and then data interpretation and feedback to the community. Because if you don't do it, then we just become, what do they call them, parachute anthropologists? We kind of drop in, collect a sample, and then run out. And the people are saying, what happened there? You know? I must say that collecting a sample, maybe it's because I'm coming from the base at Howard University, which is an HBCU, but collecting a sample has never been a problem for me. People come to me, maybe I don't look very threatening, so they come and give blood, and they give saliva, and they go get their friends and bring their saliva, they bring their friend's saliva. So we end up with many more, we thought we'd get 200 samples, we got so many samples, it's been a little overwhelming. But that's what we're going to need because of the inherent level of variability in African descended populations, because that's where our species, That continent is where our species spent the most time, and it's where the majority of our mutations have accumulated. Okay, this is just, I'm castigating the work that has been done because they've had a big emphasis on hunter-gatherers, and that's cool, but it's not relevant to us because most of the African Americans did not come from hunter-gatherers in Africa. They were selected because they had agricultural skills. So, you know, and of course the statements that appear in the literature are still problematic. There was a colleague, a European genetic so-called expert on Africa who said, there's literally nothing in Africa that's not possible since we have no idea that what humans were doing on the continent 5,000 years ago. And you know, I can't tell you how upset I was when I heard that, because 5,000 years ago, we know what people were doing in Africa. Because Africa is not that, it's not a black box. It's not a big mystery. I mean, 5,000 years ago, you've got rapid and intense desertification occurring, so people are moving from the Sahara into that long oasis called the Nile, and they still continue with development of civilization. And so you see the Nile Valley populations receiving all these migrants from the Sahara, as well as from upper Egypt, and you get settlements. And you know, so 5,000 years ago was not that far back in African history. We just don't know. And what did I say at the physical anthropology meetings? I said, genetics without history is just rumor mongering. Like you're just throwing out a rumor about where you think people are from. If you have no sense of their history, and I'd add to that a sense of their cultural priorities, because everybody's got a history. This is just some of the ancient DNA, which is the collecting petrous bone and tooth samples from the skeletal collection produces what's called ancient DNA. As you can see from this map, most of the ancient DNA studies have been done in Europe. So we know a lot about ancient DNA in Europe, very little about ancient DNA in Africa. But once we can get that going, you know, it's going to be very revealing. I mean, it will change many of our paradigms about who we are as a species. This is just more about the migration patterns. As I said, we've started analyzing our data already, and we know that sometimes in UAE, for example, when we're looking at the Red Sea migrations, we're picking up African mitochondrial variants in Arabs from the United Arab Emirates, but the places where those African mitochondrial variants would have come from, they're no longer there in that part of Africa. So people are constantly moving around Africa, which has led us to realize that the most important migratory event in African history has been intra-African migration. You know, the most important admixture event in African history has been intra-African migration. These are just the principal component analyses. If you've seen these where they show the genetic distribution and they lay on top of that the map of Europe, and it pretty much matches up with the map of Europe, meaning that geography rules in the distribution of genetic variation in Europe. We'd like to know that if that's the same case for Africans. Or here's one where they took Europeans from the transatlantic diaspora in Colombia and Venezuela, and they were able to match them back to particular communities in Eurasia, in Western Eurasia, especially in Spain and in Italy. So that's, you know, we'd like to do that as well. In fact, we'd like the same kind of capabilities that are available for European populations, genomically, to be available for African populations. And that's not too much to ask. We've got the technology, we just need the support to do it, you know? And then we'll create a database that everybody can use, which should, you know, it's gonna challenge some of the old assumptions that are out there, but that's the nature of science, right? That's what we're supposed to be doing. Okay, so this is just me saying, again, all the things that we need. Investment in infrastructure, of course, continued support from the African descended community, And of course, we need our partnerships. And these are some of our collaborators. We have a lot of people working with us because we need a lot of help. And that's also the nature of our existence, that no one gets through this science by themselves. If we want to implement it and make it real for the population, we have to have many partners and many hands out there. So I thank you all very much, and I hope that this has been interesting for you.